Welcome to Study Loop.
We are being asked to compare and contrast prokaryote and eukaryote gene expression. So real quickly, let's look at prokaryotes. Prokaryotes have a circular genome, and throughout that circular genome, you have different genes um, dispersed throughout. And over here, I've drawn one exaggerated version of that. In the green is the gene that encodes for a protein, and in the red, you have the promoter region or the operator region. And to that promoter or operator, transcription factors or repressors or activators can bind, which can then lead to the transcription of the gene. So suppose that we have all of the correct activators and transcriptional factors in place, and all of the repressors for this gene have been removed, then RNA polymerase can bind to that gene. It can start making RNA, and while that RNA is still being synthesized, while it's still being transcribed, a ribosome can bind to that RNA and start making protein right away. And that's what referred to as uh, co-transcriptional translation. So translation is happening at the same time as transcription because there's no division of space of nucleus and cytoplasm in prokaryotes. And the only level of regulation happens at the level of the regulatory region on the gene, which is promoter or operator region by the binding of transcription factors or repressors. Similar in eukaryotes, you have the gene that is being transcribed, and you have a promoter slash regulatory region. To that promoter or to that operator, different proteins can bind, and those proteins can be repressors or activators or transcription factors that modulate the transcription of the protein. Once the gene is transcribed, you have pre-mRNA. That pre-mRNA gets processed into mature mRNA, messenger RNA, through the addition of 5' caps and poly A tail additions, through splicing and through the binding of RNA binding proteins. And then once that once that mature mRNA is produced, it can be translated into protein, and then the protein can be further modified through the addition of chemical groups like phosphate groups. And just so that it sort of sticks a little bit more, since I haven't used blue, let me use blue here. You have in this step here, you have transcription. So what I've drawn here, that is a chromosome, like an X chromosome, and you pick one gene from the chromosome, you transcribe it into pre-mRNA, you process the pre-mRNA into mRNA, and then the mRNA makes it out into the cytoplasm. I'm sorry for the double arrows. I'm not really sure what I was thinking at that moment, but I hope you can follow. So the mature mRNA, after processing, makes it out into the nucleus, and then that's where translation happens. Whereas because in prokaryotes you do not have that division of the nucleus, that is not there, the ribosome can bind to the mRNA right away and start translating it into a protein. Okay, so now let's look at our answer choices and see which one is correct. So choice A, prokaryotes show co-transcriptional translation. Yes, that statement is correct. Whereas eukaryotes perform transcription prior to translation. Yes, that is correct, and that is what we're seeing here. Transcription happens in the nucleus, translation happens in the cytoplasm. In both cell types, regulation occurs through the binding of transcription factors, activators, and repressors, and that is correct because in both cell types you have regulatory regions where proteins that either promote or repress gene expression can bind to. So choice A is completely correct. Let's see choice B. Prokaryotes perform transcription prior to translation. It happens at the same time, so that's wrong. And then the second part of that is also wrong because it says whereas eukaryotes show co-transcriptional translation. So both statements from choice B are wrong. Choice C, prokaryotes show co-transcriptional translation. Correct. That is regulated prior to translation. Well, then that doesn't make sense, because if translation is happening at the same time as transcription, there is no before, they're happening at the same time. So it's saying that co-transcriptional translation is being regulated prior to translation. You see that is self-contradictory. That doesn't make logical sense. So choice C is wrong. Choice D. Prokaryotes show co-transcriptional translation that occurs in the nucleus. Well, that is wrong. Prokaryotes do not have a nucleus. So the clear correct choice in this question is choice A.